Thank you, Terry. I'm joined now by Basar Bilgicer, an associate professor in the Department of Chemical and Biomolecular Engineering in Notre Dame's College of Engineering, so he's a real dummy. He came to Notre Dame in 2008 after finishing a postdoctoral fellowship at Harvard University. His research is in the area of biomedical engineering, specifically designing solutions for complex diseases like cancer, autoimmune diseases, and allergies. So, Basar, welcome to Notre Dame Day.
Thank you for having me.
Great to have you here. When we talk about these diseases, we often think of cancer, and automatically we assume that it's a problem that's going to be solved by the medical community. So, where does an engineer fall into that?
Well, engineers have been working in medical field for a long time, actually for over a century, but most of it has been on the side of instrument development for diagnostic applications. With the knowledge becoming available on the molecular level, with the biology of the cancer cells, the research that is being conducted by basic scientists, basic science researchers at places, at cancer centers like our Harper Cancer Research Institute here, engineers like myself can now actually design molecules to overcome cancers and target allergies and other diseases and conditions.
Now, we all have loved ones who've been affected by cancer, and we know what chemo can do to the body. I mean, yes, it can kill cancer cells, but it can ravage the other ones as well. Are we getting closer to a solution for this problem?
Well, we've been working on the development of nanoparticular drug delivery systems. And these nanoparticles, unlike the chemotherapeutic drugs, when the patient takes it, it basically gets distributed through the body to all tissues non-selectively. About only 0.5% of the drug gets to where you want it to go. Yeah. By packaging it into these nanoparticles that are much smaller than the width of a hair, we can actually prevent them from hitting the healthy tissue, and while having them accumulate at the tumor site. Now, we also cover the surface of these nanoparticles with targeting agents that are like harpoons to attach themselves to the cancer cells. These smartly designed particles then are taken up by the cancer cells like Trojan horses and then deliver their lethal dose into the cancer cells, improving the efficacy of the drug, while all at the same time reducing the systemic toxicity, basically the terrible side effects that we all know about.
Okay.
And
go ahead.
Well, we've been working on developing these systems, and we do the cellular basic sciences research, as well as we're working on animals. And during these preclinical studies, we got some really exciting results that we're very eager to take to the next stage.
That is good to hear. So are you nearing clinical trial stage?
We are ready for clinical trials, in fact. However, the jump from an academic lab to taking things to the clinic is not that trivial. It's usually, we cannot basically inject the molecules that we make in our labs here at Notre Dame into humans. The FDA regulates this very tightly, which means that we need to start a company or companies that will use and facilitate the clinical studies. In fact, a group of my students at TUM that I have been advising used our technology to enter the McCloskey business plan competition, and they beat 123 groups, and they became first. They won the competition, and now we're in the search for funds to get the company up and moving.
Boy, that's wonderful. I understand you're also researching diagnostic methods and drug development for serious allergic reactions, both drug and food. Tell us about that.
Well, since we developed the nanoparticles, we realized that actually it has multiple different applications. See, the biggest challenge in the nanoparticle drug delivery field has been, because you're aware that it is not the mainstream therapy right now in the clinic, and the reason has been the synthesis of these particles, the method of formulation. And we developed a new platform that actually overcomes this batch-to-batch variation during the synthesis of the particles. And we realized that we have other opportunities to apply this technology to. And so we're now using our technology to understand allergic diseases better, because, say, someone who has allergic conditions like to peanuts, which is the most common food allergy right now in the United States, usually a peanut extract is given by a scratch method to the back of the patient or the arm to see if the patient has an allergic, will show an allergic response. But that is an incomplete diagnosis. Basically, we've developed a new method for looking at different epitopes of the allergy proteins that are within the peanut extract and to identify which one of these components do the patient actually has allergy towards. And that way we can actually develop new inhibitors specifically for those allergies without actually trying to suppress the immune system, because it's really useful for a lot of different purposes like cancer surveillance to fighting with pathogens.
Well,
So
fascinating.
it's
Thank
a lot
you
of
very
fun.
much for being with us today, Basar.